Hi all, welcome to BVW106, Introduction to Entomology and Microbiology. This subject's an interesting one where we look at some of the lesser known members of our vineyards and wineries, including the invertebrate animals, those animals without a backbone, as well as the microorganisms, those that can only be seen with the aid of a microscope individually, but we can often see their effects. That include the bacteria and the fungi, as well as viruses, and even some algae as well. And the way this subject works is we look at some of the fundamentals of these organisms, how they're built and how they operate and how they interact with each other. And importantly, we apply it. So how can we use this knowledge in a management sense? Both management of a winery, but also an agricultural system like a vineyard or any other agricultural setting as well, including, I should say, also natural settings, so true ecosystems. We often use the term in this subject agricultural system, and in that I'm referring to a modified ecosystem or a production ecosystem. And when I talk about ecosystem, I'm talking about the living organisms and their interactions with each other, as well as how those interactions are modified by the external environment, for example, the climate. The subject starts off with an overview of the invertebrate animals, as I said before, about the first 6 topics. Then we have a little test at the end of that, and then we go into microorganisms, and then we bring the two together. And then there's a short test at the end of that as well. Other assessments included in this is a invertebrate trapping exercise, as well as the analysis of what we find. That's in a major report form, which is more fun to write than taxing. There's a range of practicals that you can do both yourself and some that we do on campus here as well. And they're designed to be both interesting and useful, or as interesting as possible. As well as there's 3 oral presentations, which again aim to be more interesting than taxing. And one of those is in the form of a podcast, so a pre-recorded audio presentation, a bit like this. So in this topic here, we'll go through a little bit about the subject, what I haven't covered already, and then we'll go into an introduction to life and how we look at the division of life, the classification of life into kingdoms, species, for example, and why that's important to us. How we can use that information to do very broad mapping of ecosystems. And then we'll look a little bit at how organisms are broken up, or on an evolutionary scale, in the form of phylogenies. So I've been throwing out a lot of new terms here that might be new to you, like phylogeny or even ecosystem. But we'll be going over these topics and these words and these concepts a range of times to get our head around them. We'll be applying them. So don't worry if I'm throwing out new words really early on here on the first slide, because we'll get plenty of opportunity to go through them as the topic, or as the subject, develops. So here's the subject's objective, and pretty much as I said before, a theoretical and practical knowledge of invertebrates and microorganisms. And microorganisms are also invertebrates, and they don't have a backbone. So when we say invertebrates here, we mean animals without a backbone. And the whole central philosophy is understand why things are to deduce what they are. So instead of remembering long lists of insects and bacteria and their names and their key characteristics, what we want to get is general rules that we can apply to specimens we might find without even knowing what they're called. But by understanding why they evolved to have certain features, we can start ascribing them particular potential functions that they may play in the agricultural system we're looking at. Professional standards, of course, and at this point I should point out the subject outline is found on Moodle, and it goes through the full topic list as well as the assessments in detail. There's a separate assessment manual as well, which we're building on, we're adding pracs to. The first practical is contained in it already, as well as a short style guide, as well as the professional standards including plagiarism, All assessments are submitted via the Moodle page. Professional standards, as outlined there, are fairly obvious, and they're exactly the same as what you'd expect from me. So hopefully I can keep them, and hopefully you can keep them as well. So there's a quick quiz that we normally go through, but what I've done is I've put that into a short Moodle quiz at the top in topic 1. And it's just to see where you're at. Well, it's more fun than anything. And there's 3 questions in that. So I'll point you towards that at the end of this presentation and just click through that. And the answers are fully worked out. So even if you don't get it right at the start, which is fair enough because it's topic 1, you'll still be learning something from it. So entomology is generally referred to the study or the science of insects. Here we use it as the study of invertebrates because inevitably when we're talking about insects in an applied sense, in an agricultural system, we inevitably encounter animals without a backbone that aren't insects. So insects are a type of invertebrate animal, but not all invertebrate animals are insects. So it's a bit limiting if we only looked at insects because from a viticultural sense and as well as any other agriculture, we have a lot of invertebrate animals that aren't insects that are important to us. That's a very roundabout way in saying that in this subject we'll use entomology to describe the study of invertebrates as a whole, not just insects. And the first question of the mid-semester test is always, what is the definition of entomology? And the correct answer for that, for the benefit of this subject, or in the context of this subject, is invertebrates, the study of invertebrates. Microbiology is the study of microorganisms, which are defined loosely as those which can only be viewed under a microscope. That's a traditional definition of microbiology. But of course, as time goes on, the edges of this field gets blurry, and there's some microorganisms that we can actually see without a microscope in rare examples. But pretty much the field of microbiology has shadowed that of the development of microscopes about 400 years ago. And when we go into microbiology, we'll go through the interesting evolution of microscopes very briefly. The amazing scientists that led the way there. And you think about it, before these microscopes, even these primitive microscopes were developed, invented, we had no idea that there was such thing as bacteria, viruses and fungi, and we certainly had no idea that these were the cause of a lot of the diseases that inflicted the human race. So we'll start now by looking at life as a whole and how we divide it up and why these divisions are important and what we can learn from that. The broadest possible classification of all life on Earth is the level of kingdom. It's the biggest pigeonhole that we can put groups of organisms, and traditionally we've put them into 5 large pigeonholes called the kingdoms: Monera, Protista, Fungi, Plantae, or the plants, and Animalia, or the animals. Now, this classification system again is more complex. It's developing all the time, and there's different schools of thought as to how it should be done. But from an applied sense, applying it to agriculture like we wish to do, the 5 kingdoms are well and truly fine for us. So what we're going to look at is, or remind ourselves of, how are organisms divided into these 5 kingdoms, and then how do we divide them more specifically below these kingdoms. Where did this system come from? Well, it came from the great Carl Linnaeus, the Swede, born in 1707 and passing away in 1787. He was a true Renaissance man, did many great things for science and the world in general. Probably no greater than his legacy will always be viewed as based on his hierarchical system of taxonomy or classification, where he went, okay, let's decomplicate how we classify things, how we pigeonhole things, and give a strict order on how we do it, going from really broad categories, kingdom being the highest, down to really specific categories, species. There's lots and lots of organisms found within a kingdom, but there's only one type of organism found in the strata of species, at the level of species. I like to think of his hierarchical classification, system of classification, as addresses. So our address can be extremely specific in that it could be a particular room at a particular number on a particular street in a particular suburb in a particular city in a particular state in a particular country. That's very similar to the classification system. So here we have kingdom being the broadest group, then phylum, class below that, order, family, then genus, then species. If we looked at that in address terms, going from the kingdom level down to species, we'd have country as kingdom, phylum as state, class as city within that state, order as suburb within that city, family as a street in that suburb, genus as the number on that street, and species being a particular room or building at that number on that street. So for example, my office here would be room 116, number 60 Cooper Street, Epping, Melbourne, Victoria, Australia. And when you think about addresses, there's lots of different houses. There's lots of houses in Australia, so it's a very broad way of pigeonholing a house. Then there's less houses within the state of Victoria because there's other states. Less houses again in Melbourne because it's one city within Victoria. Less houses again in Epping because there's a range of different suburbs in Melbourne. There's a whole heap of streets in Epping, and there's a whole lot of houses along Cooper Street. Likewise, there's a lot of different rooms at this particular address. So that's how the hierarchical system of classification or taxonomy was developed by Carl Linnaeus, and it was a universal system, which is important as well because before Carl Linnaeus came up with this system, different countries or even different scientific groups in the same country had different methods for classifying organisms, and that was extremely confusing, especially when you were having collaboration across country borders. The first system was developed by Aristotle about 2,500 years ago, and he divided all life into either containing blood or not containing blood, extremely broad classifications. And the problem with that is some things with blood are very different from each other. For example, the sap in a plant could be considered blood, as would be the blood which is contained in our veins. And so putting plants and animals together is probably too broad a classification or groupings. So the step that Carl Linnaeus took was a big one, and the one that we still use to this day. So a good way of putting this perspective is, how do we classify humans, or how do we classify all life? Well, in these particular groupings, we gradually partition and subdivide life depending on traits that they share with each other. And so here we have a worm, a gorilla, or an ape, and humans. And we know that apes and humans are more closely related than, say, humans and worms. And that's quite clear because they have many anatomical features which are quite similar. Here's an exercise we would do where we reorder. Here on the left are the official levels of classifications of humans, and on the right are the description of what that level of classification means. So for example, Animalia, which is the kingdom, of course, is the top level of division of life. And we're an animal, of course, and animals are referred to as defined as multicellular organisms with more than one cell that are eukaryotic, or in other words, they have the DNA, which is in each cell, contained in a little membrane-bound sac called the nucleus. So let's have a look at how humans are classified. So kingdom, Animalia, we know this. Multicellular organisms that are eukaryotic. Again, eukaryotic means the DNA that's in each cell is nice and packaged up in a membrane-bound nucleus within the cell. The next level down, phylum. We're in the phylum Chordata, possessing a backbone. So all organisms within the phylum Chordata, kingdom Animalia, are multicellular organisms that are eukaryotic and possess a backbone. Now there's a number of different phylums within Animalia, and so the other phyla would include all the organisms that don't have a backbone. So for example, insects, they wouldn't be found in kingdom Animalia, phylum Chordata. They'd be in kingdom Animalia, phylum Invertebrata. Underneath that we have subphylum. We're invertebrata, backbone with vertebrae, and we're bilaterally symmetrical. We'll go through that in a minute. So our backbone is divided up into individual units called the vertebrae. Underneath that is class. We're in class Mammalia. It means we're endothermic animals with a backbone, with hair, mammary glands, and we give birth to live young. So all mammals within this class have those same features, and all the features in the classification levels above that. Of course, we know there's a lot of mammals, like there's a possum, so clearly the possum would divide off somewhere beneath the level of class. Maybe it's the level of order. Let's have a look at this. The order we're in is the order primates, organisms with 5 fingers, upright posture, large brain, stereoscopic vision, so 2 eyes, and feet and hands that have different specializations. So it looks like, say, a possum and us, we're in different orders. We diverge off into the primates order, whereas possums diverge off into whatever order they're in. We go beneath order, we're getting quite specific now. So we're down to the genus level, which is the second most specific level we can classify things at. And we're in the genus Homo, or human. And then our actual species name is Sapiens. So we're in the Linnaean system of classification called by the last 2 levels of classification, the genus and species. So we're Homo sapiens. There's been a number of different organisms within the genus Homo, not just us, but many of our ancestors. And even at one point there were 2 different species within the genus Homo on Earth. And there's some great literature that's easily found on the evolution of humans. And you can read about how we got to where we are. For example, from our ancestors Homo neanderthalis and Homo erectus. Think about the grapevine. The grapevine, genus Vitis, species Vinifera. There's a number of different species within the genus Vitis. There's Vitis rupestris, Vitis riparia, Vitis berlandieri. They've all, through evolutionary time, diverged away from Vitis vinifera, the grapevine that we know and love. Therefore, the question is, what is the definition of a species? Well, the true definition of a species are the members of a species are able to intermate and produce fertile young. So young that can also produce fertile young, or young that can reproduce. Therefore, Vitis rupestris and Vitis vinifera couldn't breed naturally, theoretically, and produce seed which would then make viable young. So that's why they're put in different species. So each organism, as I said, is given their genus and species name for short. We're Homo sapiens. Scientists used to get the opportunity to name organisms that they found themselves. And so, for example, one of the more humorous cases is a scientist who was trying to find an insect for a long time, finally found it, so he put it in the genus Agra, species Vation, aggravation. It's about as nerdy as it gets. Nowadays, and often scientists, of course, name them after themselves, but nowadays there's actual boards of review which allocate names. You generally don't get to do it yourself if you're lucky enough to find a new species. And new species are being found as we go through in topic 2. Even new genuses are being found, and occasionally even new orders, whole new orders of organisms are found. Alright, let's look at these kingdoms individually. Firstly, we have Monera or Monera. This includes all prokaryotic organisms. So we looked at eukaryotic organisms before. They were organisms with the DNA found in a membrane-bound nucleus. So a little sac inside the cell. Prokaryotic organisms. So these ones, they don't have their DNA found in a little nucleus. Their DNA just floats around inside the cell. Anyway, there's just one type of prokaryotic organism, and they're the bacteria, and bacteria are those that make up the kingdom Monera. They deserve to have their own kingdom because they're the most primitive organisms on Earth, and everything has evolved from the bacteria that came around about around 3.6 billion years ago. Nowadays, there used to just be the kingdom Monera, and they had the one type of bacteria in, and then about 30 years ago, of course, some smarty pants happened to find that there was 2 broad categories of bacteria actually. There's these RK bacteria, which don't have this sugary substance called peptidoglycan in their cell walls, and the U bacteria that have peptidoglycan in their cell walls. And they've discovered that these new RK bacteria they didn't know anything about are actually the most predominant or abundant type of bacteria on Earth. So nowadays, these RK bacteria and U bacteria are actually found pretty much in separate kingdoms. But again, for our applied sense, we don't need to go much further into that. The protists are another kingdom which of historic significance, but becoming less prominent now. But this is a kingdom which will lump all organisms that we don't put into the other four into. They're always eukaryotic, because there's only one kingdom with prokaryotic organisms, that's the monera. So the protists are the simplest types of eukaryotic organisms. And most importantly to us, it includes algae. It includes a range of other organisms as well, including these beautiful diatoms, which generally contain silica. And they make up a bulk of the phytoplankton found in the ocean, which is one of the most important sources or primary producers in the ocean. Primary producers being those organisms that bring the energy into that system. If you go on YouTube and look up diatoms, you'll see all sorts of amazing, beautiful pictures of them. Another interesting protist is the plasmodium, which is found within the mosquitoes, which cause malaria. So protists can be single-celled or multicellular, and the multicellular ones are quite simplified and have very little specialisation that we see in a lot of other organisms. Fungi are another interesting kingdom. I'm probably going to say that about every kingdom because they're all interesting, but fungi are an amazing one. They can be single-celled or multicellular, and sometimes they can be single-celled and then turn into multicellular, and then convert back to being single-celled. We'll go more into that when we get to that topic. Single-celled ones we generally refer to as yeasts. So the yeast that we know and love, including Saccharomyces cerevisiae, which we use in the winery all the time, is a single-celled form of yeast. There's also multicellular types, and there's a picture of one there. They include the mushroom, which are generally quite delicious. These are eukaryotic, so they have their DNA in those membrane-bound nuclei in the cell, those little sacs inside the cells. And they have cell walls. Animal cells don't have a cell wall around them. They have a membrane, but not a cell wall. Fungi have a cell membrane, which encloses their cells, but as well as a stronger wall around the cell as well. Plants have these cell walls, but the cell walls in plants are composed of cellulose. In fungi, on the other hand, they're composed of chitin. Now this is interesting, chitin, because chitin is the predominant substance in the skeletons of insects. And this gives us a clue about the evolution of life, because it tells us that it looks like fungi are actually more closely related to insects than they are to plants. Even though insects don't have cell walls, that doesn't matter. They both contain this substance. Another thing which is similar between the two is that fungi are sort of quite animal-like in the way they eat. They actually release enzymes into the surrounding environment, break down organic matter, and absorb it through a form of root system called hyphae. Again, we'll go through that in great detail in the fungi lecture. Fungi can either be saprophytic, that they feed on dead tissue or dead organic matter, or parasites, they feed on live organic matter. Animalia, important to us because we are one, are mainly marine because most of the earth is actually water. So it makes sense that most animals are actually found in the water, as well as we originally evolved from water-based organisms. They're heterotrophic, so they're unable to photosynthesize or use the sun for energy, unlike plants and algae. And also some types of bacteria can as well. They're ingestive in that they move around, find food, and bring it into their internal environment. They don't produce that energy in their internal environment, they ingest it. They're always multicellular, and their individual cells are grouped together into tissues, and those tissues are grouped into combinations of tissues which produce functional units called organs, like the heart and the brain. So kingdom Animalia, we'll burrow down into this kingdom Animalia now. It's divided itself into little subkingdoms. Subkingdom Parazoa includes the sponges. Here's a picture of a sponge here. And these are animals that are not symmetrical. And here's symmetry, just as a reminder. Top left here we have a sphere. And a sphere can be symmetrical on a number of different planes, referred to as radial symmetry. Here on bottom right we have a bilaterally symmetrical object. It's only symmetrical down one plane, down the middle, and that's what humans are, of course. So we're in the subkingdom Eumetazoa, all animals that are bilaterally symmetrical, or symmetric, or have symmetry, either radial symmetry or bilateral symmetry. We, of course, are bilaterally symmetrical. So at this point, even with this information we've armed ourselves with about the different kingdoms, we can actually start applying it in a crude sense. So if we think about an ecosystem, ecosystems are simply groups of organisms interacting with each other, and in some shape, way or form transferring energy between them. And this transfer of energy is really what all biology is about. How does energy transfer from a non-living state, like photons from the sun, into a living format in the form of sugars or organic matter? And so here we have a very simple ecosystem I've represented by simply three kingdoms interacting with themselves in some shape, way or form, here represented by an arrow. So potentially, if the arrow represented eaten by, this would say kingdom 1 is eaten by kingdom 2, which is eaten by kingdom 3. And so if this was the entire ecosystem, then we could actually limit what kingdoms could be found in each spot. If this was the extent of the ecosystem, then kingdom 1 would have to be what we refer to as a primary producer, one of those things that brings energy into an ecosystem. Because if there was nothing bringing energy into an ecosystem, it couldn't function. So, in other words, kingdom 1 would have to be something photosynthetic, like a plant or an algae or a photosynthetic bacteria. And then, if this arrow represents eaten by, let's say it's a plant, kingdom 1. Kingdom 1 is eaten by kingdom 2. What could that be? Let's say an animal. Let's say a herbivore. So we've got plants eaten by herbivore animal. An animal, brackets, herbivore. And this herbivore is eaten by, let's say, another animal. That would be a carnivore. So now we've got plant eaten by animal, eaten by animal. And not only are we saying, animal in kingdom 2 and animal in kingdom 3, we've actually we've further qualified what they are. We've got herbivore as kingdom 2 and carnivore as kingdom 3. That's the only way that system would work. If we change that arrow to lives in, it would be a range of different kingdoms could be in these different kingdom 1, 2 and 3 as well. So the best way to go from here is to get an agricultural system that you're familiar with and make a chain of interactions between organisms that are found in that ecosystem. And what is bringing energy into that ecosystem, and what would these red arrows represent? Eaten by, or lives in, or lives together. So have a go at that, have a think about that during the week. What is missing from the figure? Well, we've added that, I talked about that before, is where does the energy come from and in what shape, way or form? If that was sunlight, another arrow there, then that would have to be plants. Alright, so we've looked at animals and we've seen that they're divided up based on their symmetry, essentially, which is strange when it's the first time you hear it. I can't believe life is divided up one of its highest levels by if it's symmetrical or not. And then we're going to go further down the animals, into the symmetrical animals, looking at those without a backbone. So it doesn't include us, remember, because we're in chordata, we have a backbone. So we're going to look at the other phylum, the ones without a backbone. the invertebrata. And there's a range of organisms within this phyla, including the nematodes, which are the roundworms, the mollusks, clams, oysters, snails, and squid. And there's an interesting grouping: snails with squid, squid with oysters. So they all share. Particular traits. Then there's the annelids, or the segmented worms, including the earthworm. So if you think about those earthworms you pick up in your hand, they got those ribbings or ridges on them. They represent different segments, developmental segments of that organism. And this is an important evolutionary step that life made, this segmentalization, because segmentalization allows for specialization. So in each segment of that organism is able to specialize. And this is why annelids and nematodes are in different groups in this one phyla, because nematodes are a lot more primitive and they don't have this segmentalization. So we find that nematodes are much more simpler in structure than annelids or the worms could possibly be, because they don't have this segmentalization. Arthropods, we're going to be burrowing down into a lot further. Arthropods all have the same structure, and they include a whole range of organisms: crustaceans, insects, and arachnids. So classes within arthropoda, the arthropods, we have crustacea, chelicophoras, spiders, scorpions, and mites. Note that mites are not insects, although we will be talking about mites, and that's why you can't just use entomology nowadays, I think, just for the study of insects, because you're not going to ignore mites in your vineyard, for example. Myriapoda, millipedes and centipedes, again, they're not insects. Hexapoda, which are the insects, which we'll be going to now, and an extinct group, Trilobitomorpha. So these are all classes of arthropods. What is an arthropod? Seen all these different classes, but what is an arthropod? Well, arthropods all have the same set of features. First of all, they have a hardened, jointed exoskeleton composed of chitin. Remember that stuff I talked about with the fungi? It's this sugar-containing hardened substance. And if we have a look at its structure, we see it's got these sugar molecules. We won't be going lots into chemistry, I can tell you, in this subject, but here we have a sort of a basic sugar molecule in the middle with just these functional groups containing nitrogen. And it's these modifications which allow the sugar molecules to be strengthened into an exoskeleton. This exoskeleton, because it's on the outside of the organism, if the organism grows, it has to remove this exoskeleton and grow a larger one. Or slow off the exoskeleton, or molting, of course, is the word I'm looking for. So all arthropods have these exoskeletons, a skeleton on the outside, and it's composed of chitin, which is based on the sugar molecule with these functional groups with nitrogen in, which makes it hard. We'll be looking at the exoskeleton in a bit more detail next topic, and we'll see what features of it are important from an applied sense. All right, so they've all got that. And then they've got another structure as well, or key characteristic, I should say. And that's they're broken into a set of 3 tagmata, or large segments. The head, the thorax, and the abdomen. Head being at the front, of course. The abdomen in the middle, the equivalent of our chest. And the abdomen's everything else at the back. Arthropods have mouth parts, which used to be legs, but they've evolved into mouth parts of differing function. Their legs have segments in as well, which allows them to be jointed and allows them to move, have articulation. Appendages in arthropods are always pairs. They've always got pairs of legs, for example. Arthropods have eyes. They're either simple eyes, which are single optical cells, or compound, a whole lot of optical cells joined together. And here's a classic microscopic image of a fly's eye. See all these little individual units? They're all individual optical cells. The more optical cells they have, the better their vision. That's simple. Arthropod anatomy, we look here now at respiratory system, circulatory system, and digestive system. All of them are really just simple tubes. Unlike our respiratory system and circulatory system, which is sort of complex and muscular, and branched,
The arthropod anatomy is branched, internal anatomy is branched, but they're really just sets of simple tubes. The gut is just a tube that runs from the mouth to the anus with limited specialisation. Most of the specialisation is found in the crop area for breaking down foods if required, and down further on in the digestive or the absorbative part, the hindgut, where there's things called Malpighian tubes, which prevents excess water loss. We'll go into that next week. The respiratory system is a whole lot of open tubes called trachea, which open up to the outside environment via these little holes called spiracles, which resemble the stomata in plant leaves. And here's an example, microscopic image of the spiracle. See, that's on the outside of an insect, and it just allows oxygen to diffuse into the insect. The circulatory system isn't like in us, where it carries oxygen around, as well as nutrients and hormones. Instead, it just carries nutrients and hormones around. Oxygen is carried around only by the respiratory system. The circulatory system also removes waste products from muscle areas and the like for removal from the body. Here's a circulatory system here found on the dorsal surface of this arthropod. Underneath it on the dorsal surface and also down the ventral surface, we find trachea making up part of the respiratory system. The heart is really just a sort of muscular part of the tube which helps pump this blood around the body. And remember, it's not blood as in oxygen containing blood. It's actually referred generally as hemocoel, Hemocoel, and it doesn't carry oxygen. The nervous system is just a whole lot of nerve cells joined together. The brain is a whole lot of them fused together. But what it forms is a very reflex-orientated nervous system. It goes from a sense organ, say the antenna, directly to where an action has to take place, and short circuits the brain. So they don't get a stimulus, think about it, and react to it based on their thought. It's generally more primal or reflex-orientated than that. So, for example, their antenna may pick up a particular compound that they are attracted to. That will send a signal to the wings and it'll make them fly towards it. On the other hand, if the chemical signal is something they're repulsed by, they'll fly in the opposite direction. So it's a very reflex-orientated activity. And we all actually take advantage of this sort of reflex-based nervous system with some of our management strategies, believe it or not. Arthropod reproduction, generally based around egg laying or ovipary. Some arthropods don't require mating to reproduce, and they reproduce asexually, so they produce little clones of themselves, but this is rarer than sexual reproduction, which is found in most arthropods. And generally, the juveniles of arthropods don't resemble the adults. They don't even resemble a little small version of the adults, like in humans. We'll examine this further later too. Alright, so that's the arthropods as a whole class, as a whole group. They have an exoskeleton, and their body is composed of a head, thorax, and abdomen. Now we're going to look at a particular class of these arthropods, the insects. And these insects have an exoskeleton, they have these three broad segments, but they also have their own characteristics. They have a pair of unbranched antennae on their head. They have the three body segments, of course, but they have three pairs of legs attached to the thorax. Only ever three. They have compound eyes, not just simple eyes. They have the compound eyes, a whole lot of those optical cells joined together. They have one or two pairs of wings on the thorax, and they're the only arthropods that can fly. And they have chewing, sucking, or sponging mouth parts, which we'll be looking at in detail in the next topic, especially how we can use that information. Alright, so next topic we'll be looking specifically at the insects. Alright, just to finish off here, I want to touch on a way of visualising the relatedness of organisms, which is important if you're going to be reading more about them, which you will be now and in your professional life, and that's phylogenies. Phylogenies are family trees when we're looking at broad relatedness between different species. Family trees we generally use to look at relatedness within species, so like which we'll see an example of next. Phylogenies again on a broader scale. And these are used in an applied sense, and we'll see a couple of examples of those later on. And so these phylogenies, family trees generally over hundreds or so years, few generations, whereas phylogenies look at divergence of organisms over millions of years. So here's an example of a family tree with grandparents and all their kids and then the next generation below that. But here's an example of one over millions of years. On the left, we have the branching phylogeny or family tree of a group of bacteria called Brucknera. They all diverge from, see there's the arrow there, that's pointing to a common ancestor all these bacteria had 250 million years ago, or 150 to 250 million years ago. And in the time since then, they've diverged into 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 different species, all of Brucknera. From a single ancestor, they've branched out. Interestingly, if we line up this family tree of the Brucknera bacteria, it closely aligns with a similar family tree of aphids, the insect aphid. And so if we line up the phylogeny of bacteria over 250 million years with that of aphids over 250 million years, they mirror each other perfectly. And that's interesting, and that shows us, it tells us that they must be related in some way, and in fact they are. These Brucknera bacteria have to live in aphids to survive. Likewise, the aphids must have this type of bacteria within them to survive. Now we can use this. We can use it to predict if we find a new aphid species, we know that there must be a bacteria in it. Also, if we think about what conditions the bacteria need to survive, we can predict what conditions the aphid needs to survive. Phylloxera is an aphid-like organism, diverged away from aphids around 200 million years ago. But because we know they're related to aphids, we can predict that they probably have a bacteria that live in them too, and they do. And so we're using this phylogenetic information to actually predict and maybe potentially manage an insect pest based on the bacteria they have within them. So these are genus or one type of organism-specific phylogenies. But then people have actually gone out and done broader ones. Here's an example on a link that you can follow to a phylogeny of all of life. Nearly every category of life that has been found has been put together in a massive family tree. And then here's a version which is a lot easier to follow from the fantastic scientific journal Science. You can read that and it's got a nice, easy to follow phylogeny of life. Worth a look. And then at the end here in this last slide, there's a couple of links to how phylogenies are being used to determine management strategies.